Good morning. Welcome to Vanderbilt's 6th annual 3-minute thesis competition. Thank you for being here. The University of Queensland started this competition as a way to challenge students to present their work compellingly, succinctly, and plainly, using 3 adjectives. This is a preliminary session. After prelims, you're all invited to join us for lunch, not with our keynote speaker. Our keynote speaker could not make it because of winter weather in the Northeast, and she couldn't get her flight. So that's a disappointment. But you all are the real event, so there are going to be lots of presentations today. But join us for lunch afterwards. Following lunch, we will announce the finalists and transition to the Board of Trust room, and that's where we'll have the final presentations, the raffle with door prizes, which you have to be there for the raffle, and the award ceremony. I would like to thank all the volunteers, the sponsors, which are the graduate school, the Peabody College of something or other, Peabody College of Education and Human Development, the English Language Center, judges, participants. Lots of people are pitching in to make this event happen, so we really appreciate all of your. Let's see. I have one note for participants. Participants, you will get mic'd before you come up to the stage. You will come and meet Chris. Where are you going to be, Chris? Right up here. And Chris will get you mic'd. Just relax as best you can, and remember that you need to wrap up your time at the cutoff. I'll say more about that in a minute. The presentations will proceed one after the other. I will call each participant to the stage. Please don't clap as participants take the stage. Instead, clap after their presentations. There will be a short pause between presentations to allow the judges to finish scoring and for the next participant to get ready and get mic'd. If you're the next presenter, come on up to Chris and get a mic. We've got 2, so we can rotate in and out. Everybody, please put your phone on silent. And the competitors today will abide by a few rules. All presentations must be presented in spoken word, so no poems, raps, or songs. Presentations must commence from the stage. They begin when a presenter starts speaking. The timekeeper will hold up a green sign, let's see it, to signify that time started, but it doesn't start until you actually start talking. Presentations are limited to 3 minutes maximum, and competitors exceeding 3 minutes cannot advance to the finals. You can still get comments, on your scores for your own benefit, but you can't advance to the finals. The timekeeper will give a warning at 30 seconds, yellow, at 15 seconds, and at time. Okay? A single static PowerPoint slide is permitted, but no animations, sounds, props, or memory aids. And we've got your PowerPoints here, hopefully. The work presented must be from a planned or in-process thesis or a dissertation, or in one case, just recently completed, I believe. It must be original to the speaker and undertaken while at Vanderbilt. Participants must attend the entire session. This session, you can go out and go to the restroom, you can go get more coffee, but you need to be here for the whole session. And if you're a finalist, you have to go to the entire final session, just so we don't have too much disruption in order to be eligible to win. To be courteous to presenters, we encourage everybody to attend the entire event, but we understand that people have other commitments they might need to make. The decision of our judges is final, so there will be no appeals of those decisions or anything like that. Okay? All right, we will now begin. Please remember not to clap as the participant takes the stage, but only after they finish. Actually, we'll begin once our participant gets mic'd. Any questions? Okay. Yes, Marianne.
Are we allowed to test the mic if we say testing to use for sound, or as soon as we say testing, it pops
That's fine. Can you just tell if people are doing that? Okay, that's fine. Any other questions? We have great raffle prizes. We have an iPad and a couple of Kindles and a couple of Keurigs.
Oh, yes.
All students are eligible for those. If you're participating or not, go out and fill out a raffle ticket. You can do that after the session too. All right, let's see. This is Joe Lusinger from Neuroscience.
Do I need to do my slide or?
Yeah, yes, I'm supposed to do that. Let's see. Okay, so this is just introing you. Joe Lusinger from Neuroscience. And this is your slide, right?
Yes. Great. I want you all to imagine someone told you to stop eating. Not for 5 minutes, not for 10 minutes, but forever. That's a pretty good analogy of what it's like when someone battling addiction tries to stop using. The processes, the emotions, the experiences in their brain have evolved over millions of years and helped our ancestors survive, but now are being hijacked by drugs of abuse. Now, the human brain is extremely complicated. It has over 100 billion neurons, and between those neurons it has connections that outnumber the stars in our galaxy. We think that those connections to each of those neurons is what gives each neuron its specific function. So understanding what those connections are doing and where they're coming from will help us understand how and why we become addicted. Now, studying the human brain is tricky because it is sort of organized like a bowl of spaghetti, if that bowl of spaghetti had 100 billion noodles. It also is a problem because people don't really like you poking around in their brain very much. And so in my lab, we simplify that problem by using mouse brains. Now, in the mouse brain, we're able to inject viruses, and these flu, HIV are examples of viruses, and these viruses that we use change the colors of the neurons so they can be identified. It sort of works like a mailman driving down the road and flipping up the flags on specific mailboxes. You can use a number of different viruses. In this case, we use rabies. And rabies is it's a modified version of what a raccoon might infect you with if it bit you. It is a unique virus in that, unlike other viruses in the brain, it can jump across those connections and flag the connected neurons. That's what we've done here. So you can see all of these green dots in this mouse brain have been flagged neurons, even though the injection where we put the rabies is just down in this bottom right corner. This is a zoomed in square on the bottom left. Once we know where those neurons are coming from, where those connections are coming from, we then want to investigate what is the function of those neurons. We can use a different type of virus and infect those neurons, which gives us the ability to turn them on by hitting them with lasers. This then allows us to investigate that function of those neurons. By investigating where these connections are coming from, and investigating what the function and how those connections work, we hope that our work, and work like ours, will help us better understand, better treat, and hopefully one day cure addiction. Thank you.
Hello, hello? Is that good? Can you hear it? Okay, cool.
Thank you, Joe. Next we have Aaron Stevens from Physics and Astronomy.
That's my title. So I'm sure we have all experienced some sort of wound in our life, maybe like a cut finger or like a scraped knee, perhaps. But when that happened, did you stop to wonder, how do cells respond to that wound? Or more specifically, how do cells even know to start healing this wound and regulate the wound healing process? It turns out that calcium plays a huge role in the wound signaling process. Calcium is kind of like the internet for cells. They use it to communicate with each other, to send information and signals, so they know what's going on around them. Without calcium, wounds do not heal properly. So my work focuses on mainly two questions. One, how does the wound determine how the calcium signals will behave? And two, how does that signaling behavior then determine how cells heal wounds? To answer these questions, we have to follow the main primary rule in the Vanderbilt physics department. Shoot things with lasers. And our target of choice happens to be innocent baby fruit flies. It's sad. The reason we do this is because laser wounds are very easy to reproduce. And we know so much about the fruit fly DNA that we can modify it to test our own hypotheses. Now, I can't show a cool video of our calcium, but I do have a picture where we see calcium flooding into cells after they're wounded. We can see the calcium because of a protein that lights up, kind of like a light bulb, whenever calcium attaches to it. So far, our work has seen that multiple effects determine the calcium signals around our wounds. For example, just a millisecond before, earlier than a millisecond after wounding, we see a bubble of hot water vapor caused by the laser that expands and collapses, which causes holes and tears in cells around the wound that calcium then enters through. Also, wounded cells send off separate signals to cells further out that weren't even damaged, and we see secondary signals that then arise. My current work focuses on using a computational and mathematical model and developing that model to further test our hypotheses, as well as gain further insight into the calcium signals around our wounds. So why is this even important, though? Well, it turns out that there are many diseases that affect the wound healing process. For example, diabetes impairs wound healing. Certain infections can arise from wounds that don't heal properly. And even certain types of cancers and tumors can be described as these wounds that never heal, where the wound healing process just never turns off and is continuously running. So I hope that my work can gather more information and insight into wound healing and wound signaling so that maybe we can alleviate and cure these diseases that affect wound healing. But it's also pretty cool to say that my job is shooting mutant fruit fly babies with lasers. So, yeah, thanks.
Thank you, Aaron. Next, we have Ibrahim Ahmed from Electrical Engineering.
Let's say I'm a robot on Mars, arms, legs, and all. My mission is to collect samples to find signs of life. Suddenly, I discover that I cannot walk. When I signal my leg to move forward, it moves sideways. Perhaps radiation from the sun has crisscrossed my wires. Perhaps a little green man has been poking around my circuits. What do? Night is fast approaching and my solar panels won't work. I cannot rely on my operators back home. It can take up to 24 minutes for signals to travel back. Can I still complete my mission? Is all hope lost? No. It turns out there is something I can do. I can learn to walk again. I use something called reinforcement learning. I take a small random action and observe what happens. I signal my leg to move sideways. Whoops, my arm just sprang up. That is not what I wanted. I make note of that. Okay, let's try something else. Let's tell my arm to move up. I jumped. That is not what I wanted either. But I did move forward. So my last action had some utility. I repeat this process several times, often choosing actions that were useful in the past. Slowly and haphazardly, I learned to walk again. This contrived example seems too out of this world. In my research, I applied the same approach to real-world scenarios. A modern airplane, for example, has hundreds of controls. What if something unforeseen happens? Something there is no contingency for? A fuel tank starts leaking, for example. Can a pilot keep the plane balanced long enough to attempt a safe landing? In my work, I was able to extend the flight time of a damaged plane by 4 times its expected life. By observing what it could do, the plane learned to pump fuel away from the leak. That gave pilots breathing room to make a safe landing. Learning from experience is so human. I want to use my research to design machines that can adapt to adversity, that can learn on the fly. So whether it's the fate of a multi-million-dollar robot, or worse, a human life that hangs in the balance, we can tip it in our favor. Thank you.
I neglected earlier to thank our judges by name. We have Catherine Hartman, Associate Dean for Clinical and Translational Scientist Development at Vanderbilt University Medical Center. Roosevelt Noble, Director of the Bishop Johnson Black Cultural Center at Vanderbilt. Tony Early, the Samuel Milton Fleming Chair in English at Vanderbilt. And Veronica Ikashoji Orlanti, a CLIR postdoctoral fellow and lecturer in the history of art here at Vanderbilt. So thank you especially for your time today. Thank you, Ibrahim. And now we have Teddy van Opstal from Biological Sciences.
As animals, we are not sterile, and we live every day associating with bacteria in, on, and around us, which we depend on for our health. This association is so ingrained that we receive our first bacteria in our mom's womb. Maternal transmission of bacteria is universal across the animal kingdom, including the simple sponge, an insect, a turtle, or a person. However, not all bacteria transferred from mom to child are helpful, and sometimes harmful bacteria can be transferred. For example, in the hen, salmonella can be transferred through the ovaries into the developing egg, which is why, like many of you, I am as terrified of eating uncooked eggs as I am standing next to sneezers during flu season. But less than 1% of developing eggs actually receive these harmful bacteria, showing that mom can control this bacterial transfer. So, what genetic tools do moms rely on to prevent the spread of potential pathogens? So to answer this question, I played a little game of genetic clue to find out where in an animal's genome is the gene that murders a bacteria's ability to harm defenseless offspring. And so, in my lab, our clue board is the jewel wasp, that little cutie in the top right corner, because it has a strictly inherited bacteria, Wolbachia, which can be transferred from mom's ovaries into the developing egg. And Wolbachia can cause some very harmful things when transferred, such as some very male-unfriendly reproductive traits, such as killing all male children, or turning those male children into females. And so, by comparing two closely related jewel wasp species, let's call them distant cousins, one cousin transfers this Wolbachia at low stable levels, while the other cousin transfers Wolbachia at high harmful levels, reducing lifespan. And so why does one cousin transfer more Wolbachia than the other? And so by doing a little genetic engineering between cousins, I was able to narrow down a gene candidate list to 32 suspects, which could regulate the control of Wolbachia from mother to offspring. And so to interrogate these suspects, I reduced the activity of each of these genes to see if this regulation of Wolbachia transfer could be lost. And through deductive reasoning, I was able to find the culprit gene, which I named Wolbachia density suppressor, or WDS. And this gene could help us, on a fundamental level, understand how moms prevent the transfer of harmful bacteria to their children. And so, I think this also can have important consequences in terms of improving newborn health or preventing outbreaks in our livestock. And so in my game of genetic clue, I know the suspect, Wolbachia density suppressor, located in the ovaries, but I still don't quite know how this gene works or its weapon of choice. And soon, I hope to finish this game, thus ending the longest game of clue in history. Thank you.
Testing, testing.
Okay. Thanks, Teddy. Allie Lutz and Fernanda Lane were not able to attend today. So next on the agenda is Harris Manning.
Inside every cell in our body, there are tiny machines doing construction work. These machines are called motor proteins. Like your average motor, motor proteins harness the energy from a chemical fuel to do mechanical work. Unlike cars and trucks, there are no mechanics to fix them. Motor proteins, they need, so motor proteins, so cars and trucks, when they break down, it's a minor inconvenience. But when motor proteins break down, they cause debilitating diseases with symptoms such as birth defects and dementia. That's why it's extremely important for us to develop tools to study motor proteins so we can learn how to build and repair them. So in my lab, I study a model motor protein called ClipXP. ClipXP is powered by the fuel molecule ATP, and it destroys old and broken proteins by cutting them up into tiny pieces. However, ClipXP is too small to see under a microscope. That's why we need to employ two techniques, fluorescence quenching and optical tweezers, in order to study them. So fluorescence quenching allows us to see an arm of ClipXP move. So fluorescence quenching uses two markers, an orange one and a black one. When we shine green light on an orange marker, it lights up or fluoresces. However, when the black marker gets close to the orange marker, it shuts off or quenches. By putting these two markers on opposite ends of an arm of ClipXP, we can watch the arm flex as the orange marker turns on and off. So now we can see the arm of ClipXP move, but how does that relate to protein shredding? Enter optical tweezers. Optical tweezers allow us to pick up objects, very small objects, using only light. we can actually watch the process of protein shredding by placing a protein on a bead and playing tug of war with Clip XP. In my research, I have combined fluorescence quenching and optical tweezers into one technique, fleasers. Fleasers allow us to see and feel an active ClipX motor. So now we can finally relate tiny motions inside of Clip XP with protein shredding. These findings allow us to figure out how other proteins similar to Clip XP work, but also this technique can be applied broadly to any motor protein inside our bodies. So finally, mechanics have the tools in order to look under the hood of a motor protein, and they can study any problem with a light touch. Thank you.
Can you hear me? Testing, testing. Is it on? Okay. Thank you.
Thanks, Harris. Next. Let's see. Next, we have Laura Addery from psychology.
So schizophrenia is a debilitating mental disorder in which people have faulty perceptions and beliefs about the world around them. So perceptions such as seeing or hearing things that other people around them maybe don't see or hear, maybe faces or shadowy figures or voices conversing. And beliefs such as that someone might be out to get them or that people can read their thoughts. Unsettling, right? So on top of these kind of social distrust symptoms that these people have to deal with, though the presentation is variable in schizophrenia, we also know that a lot of these people convey sort of odd behaviors or odd disorganized speech, which can lead to a lot of stigma from people around them and a lot of isolation and loneliness. In addition to this, the social cognition deficits have been widely studied in schizophrenia, and we know that they have difficulty with things like perspective taking, working memory, and bias updating, things that are really necessary for us to kind of navigate the social world. So because we have all these deficits all rolled into one, people have been studying kind of how to intervene with these social deficits. And so far, a lot of people have been doing social skills training and cognitive remediation training, which basically kind of just involves using a computer and pushing buttons, repeating the same tasks over and over again until you get mastery on this one lab-based computer task where you're pushing, maybe one or two buttons, right? Which is a great start, but it doesn't necessarily translate as well to like, how do I talk to someone in line for the bus? Or how do I ask someone at the grocery store a question? So what we wanted to do is create sort of a really naturalistic intervention where we train people in virtual reality so that they're immersed in a naturalistic setting. And we use a bus stop, a cafeteria, and a grocery store as our kind of environments. And we have participants come in 10 sessions over a course of 5 weeks and train on these games. So within the game, they have social missions that they have to complete. So they kind of navigate around and they find avatars to talk to, and then they engage in social conversations to earn points. We also have a narrator that gives them little bits of feedback along the way so that if they do kind of make a misstep, they know why and they know what to do next time, and then they try again. And we want to kind of increase their feeling of mastery by scaffolding this approach so that they can learn on the fly. So far we only have 18 participants. We're still working to build, but we've gotten some really promising results. So not only are we seeing increase in their social abilities kind of in lab-based tasks, but we've got sort of anecdotal reports from our participants that they feel like, I'm more comfortable now maybe trying these skills out, or I went to the grocery store and I actually asked someone a question, I felt better about it, right? So we know that this is improving their lives, and we're hoping that we'll be able to see that in the data to come and potentially make some sort of app that they could practice on their own in the future. Thank you.
Testing? Okay.
Thanks, Laura. Next, we have Sandhya Shukla from Biostatistics.
I want everyone to look around the room and think about where you would tell your friend to sit if they were to walk in at this very moment. Maybe you would tell them to sit next to you if the chair was empty, so you guys could talk. But you might not have noticed that the chair next to you actually has an unstable leg, and that the next person to sit in it will fall on the floor. This process of making decisions based on things we think are important, like your friend sitting next to you, is how we manage disease outbreaks. When researchers and policymakers meet to figure out which place should receive an intervention next, they use features like the number of people in a city or the distance to the nearest infected premises to help make that decision. But is this really the best approach? Are there characteristics that we're overlooking that might actually be important? I hypothesized that we could train a computer to manage a disease outbreak better than a human could. But rather than give the computer what I think is important, like the number of people in a city, I let the computer decide. I provide the computer with a picture, a literal snapshot of the disease outbreak, and then the computer will tell me which place should receive an intervention next. It uses an algorithm to pick apart the picture, kind of like the way the visual cortex in the human brain picks apart images. If that decision results in shorter outbreaks and lower costs, then the algorithm gets a reward. But if it doesn't, then the algorithm gets punished. Based on the prize received, the algorithm will change what it thought was important from that initial snapshot. And I keep training the algorithm until it gives me suggestions that result in shorter outbreaks and lower costs. And it turns out, my method does yield shorter outbreaks and lower costs compared to current management strategies. Well, that's great. Computers are smarter than humans. But why should I care? Because in 2015, the Ebola outbreak resulted in a $1.6 billion loss due to decreased productivity. And in 2001, the United Kingdom had to kill 6 million cattle to contain a foot-and-mouth epidemic. That's your money, your way of life affected by poor disease management. With my research, policymakers and researchers will be better able to manage disease outbreaks without as much loss and tragedy. Before I finish, I'll leave you with this final thought. Remember that you need the complete picture to make a decision, and that a computer can probably make that decision better than you. Thank you.
Thank you, Sandhya. Kuniko Hunter can't be here today, and so next up we have Elizabeth Gibson from pharmacology.
So just think of having a cough persistently for 7 days. You go to the doctor, you get diagnosed with pneumonia, you go home with a prescription for Levaquin, a quinolone antibacterial. After your 7-day course of antibacterial, you still have that persistent cough and you still have a fever. So you go back to the doctor, they take chest x-rays and take blood work, and you find out you have TB or tuberculosis. So this is a very severe infection that takes a 6-month-long course of antibacterial of a 4-drug regimen. So now you don't just have a normal pneumonia infection, you have tuberculosis, which now could possibly be resistant to that quinolone antibacterial, which could increase your treatment regimen up to 2 years. So there's only so many targets of tuberculosis. So what can we do? The tuberculosis is mutating. The drugs that are used have been used since the 1940s, and so resistance is on the rise, but still, there's only so many targets. So the focus of my work is actually looking at new drugs that target old targets. So validated targets that we know kill these tuberculosis cells, but can overcome this resistance. So my specific focus is on the target of quinolone antibacterials, which is this enzyme called topoisomerases. They help to unknot and untangle DNA to allow replication to occur. So what better thing to target than target the enzyme that allows them to replicate? If you stop that enzyme, you can stop them from replicating. So the specific drug class is called MGIs. And these MGIs, I'm trying to figure out their mechanism and if they can overcome this quinolone resistance, targeting this validated target of topoisomerases. And so what I found so far that they're more specific for this tuberculosis enzyme, and also that they can overcome quinolone resistance. So think of the MGIs this way. You are traveling to work, going down your normal quinolone road, and find that the road is blocked. What do? Do you turn back around and go home and let your work keep replicating and increasing? Or do you find another way to get to work to decrease your overall workload? You would take the MGI road, get to the same place, get rid of the work that you have to do. So the MGIs, they can overcome this quinolone resistance and can be well tolerated like the quinolones. And millions of people per year are infected with tuberculosis. So just imagine having a well-tolerated drug that can overcome resistance and help these millions of people every year. Thank you.
Thank you, Elizabeth. Next, we have Kelly Knowles from Clinical Psychology.
Think of the thing you're most afraid of. Is it spiders, cancer, losing someone close to you? What do you think is the worst part of this fear? Psychological science suggests that it's this unpredictability that makes it the most difficult for us to handle. Decades of animal research have shown the same thing. So 50 years ago, scientists brought rats into a laboratory. Some of the rats received predictable shocks that were preceded by a tone. Other rats received the same number of shocks, but didn't get any sort of warning that they were coming. The rats that received the predictable shocks were able to recover from this experience faster. The rats that received unpredictable shocks had greater negative effects on their health. But here's our problem. Life isn't predictable. However, some of us can handle this predictability or the unpredictability of our environment better than others. In psychology, we refer to this individual difference as intolerance of uncertainty. So some of us can go with the flow, others need to prepare for every possible outcome. What intolerance of uncertainty comes down to is a very basic fear, a fear of the unknown. Because we have prefrontal cortex, we have a unique ability to think and plan, but also worry about our future, which is always uncertain. Intolerance of uncertainty is linked to clinical anxiety disorders, but it also might provide a clue for how we can treat anxiety. My research looks at the relationship between intolerance of uncertainty and treatments for anxiety disorders. So imagine for a moment that you're afraid of snakes. The best treatment for phobias is exposure therapy. This has been shown by decades of clinical research and research trials. In exposure therapy, you learn to confront the thing you're afraid of. So if it's a snake, you might learn that you can actually be in the room with the snake without it biting you. Traditionally, we start with the latter. We use an exposure hierarchy. We start with smaller fears, like seeing pictures of a snake or being in the same room as a snake, moving up to our bigger fears, like touching and actually holding that snake. But what if we don't need that traditional exposure hierarchy? In my research, we're trying to change it up to better prepare us for the unpredictable world we live in. This is called variable exposure. In this treatment, we roll the die and let fate decide which step on the hierarchy we're going to attempt next. So far, we're seeing equivalent effects between standard and variable exposure therapy. And variable exposure therapy might confer an additional benefit. It helps us better prepare for this unpredictable world that we live in. So what we're learning through my study is that we have to be able to face our fear of the unknown head on. Thank you. Thank you.
You ready, Tony? All right, thank you, Kelly. Next, we have Judy Brown from Microbiology and Immunology.
Tori Kenyon was 15 years old when she was diagnosed with celiac disease, a lifelong, debilitating gluten intolerance. She tried to avoid gluten as best she could by removing it from her diet. However, if the smallest bit of gluten found its way into her cereal box during the manufacturing process, or snuck into the kitchen at the local restaurant where she was eating, Tori would be head-on with her disease. She'd be faced with diarrhea, constipation, muscle ache, headache, fatigue, and the list goes on. So for Tori and all those with celiac disease, every day becomes a battle to determine what they're going to eat. She has to constantly worry about how her food was prepared, or whether the next bite she takes might contain her greatest enemy, a single crumb of gluten. So I asked, could you imagine living this way? Could you imagine having to think about every bite you put into your mouth? What if I told you that 1/3 of us in this room are susceptible for celiac disease? Would you want to know how to prevent it? The greatest hurdle in preventing celiac disease has been understanding its cause. So for my thesis work, I've been trying to find triggers of celiac disease. And we knew for a long time that viral infections were linked to the onset of celiac disease. However, no solid scientific evidence could ever prove a connection. That all changed when we infected mice with reovirus, a common, harmless, human stomach bug. And what we found was that mice infected with reovirus responded to gluten in the same way that celiac patients did. Not only that, but celiac patients had higher reovirus antibodies than healthy individuals, indicating that they not only had a recent infection, but could have had a more severe reovirus infection. So if reovirus is our culprit, how do we put this all together? The big question, we think, comes down to timing. Children are most susceptible to reovirus infection at the same time that gluten is introduced into the diet. We think that reovirus infection causes confusion in the intestine, leading the immune system to recognize gluten as if it were a virus. And that forever scars the immune system, and gluten forever becomes an adversary. So if gluten dares to enter the body again, there are immune cells ready for battle. And unfortunately, our immune system can be overzealous in its attack, destroying the intestine and even killing the individual. So my question for you today is, if there was a safe vaccine to prevent celiac disease, would you take it? Thank you.
Thank you. Everyone, please join me in thanking our participants and judges. That concludes our preliminary session. We're done a little bit earlier than the other session because we had a couple of cancellations in here. You can go over to the other session if you want, but since there's a whole bunch of us, maybe try to do that during a break between speakers so we don't have 20 people come in the middle of a session. The next session in this room is at 10:40, so 30 minutes.